I'm really quite astounded that there's so many people here tonight. I was not expecting it. I was expecting, 12, 15 people. So thanks very much, Lindy, for organizing such an amazing turnout. It's really quite a privilege to be here talking to you today. And it's also quite, it's quite nice to see so many people interested in the kind of research that I do. So, there are lots of animals, as has been pointed out, that I've worked on previously. But I worked on the genetics of these animals, okay? So that's the common theme that runs through all of my research. It doesn't really matter what animal it is per se. It could be a cockroach or a beetle or a seabird or an elephant or a rhino. The common thread is that they've all of these animals have got DNA in them, okay? And so they've evolved. And so that's actually the main theme running through all of my research, which is it's the theme of evolution. Because all of these things have DNA, all of these things evolve, and evolution works in a relatively predictable way. And so it doesn't really matter whether you're studying baobabs or cockroaches or rhinos. Evolution works in a fairly straightforward way in all of these organisms. And so my field is actually evolution. And that, when it's evolution of a nice large mammal like a leopard or a rhino, then it has some kind of conservation relevance as well. Because what we evolutionists like to do is to figure out how much diversity is there, how much genetic diversity is there in this animal, is there a need to conserve, is there a need to do active conservation management, or is it okay to, the diversity is of a high enough level to just say, oh, well, we'll just keep them in the reserves, we'll monitor them, but we don't actually have to make any interventions. So the problem is that genetics becomes really important when you've got small populations. Populations become small, like, for example, in our rhinos, there's very few populations that are over 50. There are maybe 10 populations in the entire country which has over 50 rhinos. The rest are less than 50, which means that after a few generations, you're going to get what? You're going to get inbreeding. You're going to get fathers mating with their daughters, and etc. So, and that's inbreeding. We humans, we don't do that. We've got cultural taboos to avoid that. And it's also bad news in animals, inbreeding. So they also have their own ways of avoiding inbreeding. The problem is if we've kept 20 rhinos or 10 rhinos on one farm for the last 30 years, then they don't really have a choice. They can't avoid inbreeding. We've placed them in the same area. We're not giving them any access to any other mates. So they have to choose what's available. And so it's when populations become small that conservation and genetics especially becomes an issue. So one of the things that has been very contentious for the last, 100 years or so has been, how much genetic diversity do we have in our black rhino? Or how much diversity do we have? And how is the diversity partitioned across the species landscape? And the black rhino, as I'll be talking to you now about, is a species that is not just from South Africa, right? It's from here all the way up into all of sub-Saharan Africa, from around the Ivory Coast in the west to the Horn of Africa, and from Eritrea in the north all the way to the Cape of Good Hope. So that's an enormous range. It's pretty much all of sub-Saharan Africa. And across that massive range, it's not the same. The landscape is not the same. The vegetation is not the same. The ecology is not the same. And so those factors drive the evolution of various species. And what I'm going to talk about today is how those factors have led to differences among regional populations of black rhino. And we can talk then about that. So if I want to talk, I don't know how well you guys can see the screens. Apologies if you can't see them very well, but there's 3 of them, right? So one over there and 2 on this side. So if we take a step back for a moment and we look at what is it that we want to do with conservation? In some way, everybody in this audience is somehow involved in conservation. And the fact that you live in Hoedspruit, means that you somehow either involved or you love animals or you love the bush or whatever. Some love for nature brings you here. And so conservation biology in general, conservationists, the human beings behind conservation, generally want to all do it for the same reason, because for whatever reason we love nature and we want to see it there long after we're gone. But if one looks at what are the actual goals of conservation biology, they are to firstly describe how much biodiversity do we have. Okay, that's the first goal, and then looking at the human impacts on this biodiversity, because conservation is a human thing, guys. It's not about animals, it's about us. Okay, if there was no humans, there wouldn't be a need for conservation, right? Because the animals are doing just fine before we arrived here, okay? So conservation is a human thing. It's not a nature thing. It's protecting nature, but we need to protect nature because we're humans and we're the problem, okay? And of course, we tend to get very focused on this last point, preventing extinction, but it's only one of the goals of conservation biology. Okay, so so how much diversity do we have? What is our impact on it, and how we can protect it? All right. So on the short term, that means let's just talk about rhinos. That means preventing unsustainable hunting and habitat. In other words, stopping poaching and making more land available for rhinos. But it's just not rhinos we're talking about. It could be applied to pretty much anything. But on the medium term, right, what we would like is actually to maintain the processes that led to the biodiversity evolving in the first place. I don't know if I'm making sense by that. To manage the population such that they're not just going to be okay now, and for our kids and for our grandkids, but they're going to be okay for the next 1,000 to 10,000 years. Okay, and those are the medium to longer term goals of conservation. So, how would we manage to do that? It's not that difficult. We need to maintain large populations that have diversity, large diverse populations of whatever it is we're trying to conserve. But also, we need to figure out how to manage these populations. Because if you have a species, let's look at Southern Africa. We've got the blesbok in the north and the bontebok in the south. They're very, very closely related species. They probably had a common ancestor about 50,000 years ago, which is not a long time evolutionarily speaking. So, should we mix bontebok and blesbok? They're two different species, or the different subspecies. And the question is always, how do we manage our biodiversity? Do we mix? And if we do mix, under what situations can we do so? And this is where we often turn to genetics for a helping or a guiding hand to see, okay, is my population need an intervention? So have I got very little genetic diversity and I need an intervention? And if I do, then from where should I source the animal for the intervention? And that animal, whichever animal you bring into a population to boost its genetics, remember you've got to make sure that that animal breeds in the population that you've moved it to. Because if it doesn't breed, then your intervention is meaningless. Okay, so you've got to make it, take care of all of the social, the ecological factors whenever you're thinking about an intervention for genetics. Okay, because just moving an animal to a new reserve doesn't mean he's going to breed there. And you want him to breed, or she, right? So the first of these medium to long term, the first basically means that to do the first part, we need to figure out how much genetic diversity we have. And the second part is to see how that diversity is structured across the landscape. And I know it's the first big word I'm using, it's structure. No, evolution was not a big word. Structure is the first big word I'm using. So structure, and I will use this again and again in this talk, but structure, it basically means how is the diversity structured? Are there pockets of diversity that are not in any other place? In other words, if you just look at us humans, we've got a lot of diversity. Just look at our skin color, how diverse we are in our skin color, right across the world. That's biological diversity. And is that structured? Yes, it is. In the northern part of Europe, you have people with a paler skin. In the more tropical parts of the world, you have people with a darker skin. So the genetic biodiversity, even in humans, is structured. It's not that you got everything everywhere, right? And that's what I mean with genetic structure. Is all the genetics everywhere? No, it's not normally. Normally there's a river or there's a mountain or something that prevents animals from passing, and then you get two different populations or subspecies or eventually even species evolving in that area, in those two areas, because there's no way for them to exchange genes. Okay, so that is what I mean by structure. So how do we figure out how biology is structured, biological diversity is structured? The old way of doing it is through taxonomy, so subspecies, and everybody knows the subspecies something copensis, right? Because that's our Cape subspecies. But you'll find with most animals in Africa, whether it's birds, mammals, you name it, we have got at least one or two or maybe even several subspecies that are peculiar to our southern part of Africa, right? Compared to Central and East Africa. And so that's already telling us that for a lot of species, South Africa, the population in South Africa, or maybe even Southern Africa, is different to the populations elsewhere in Africa. And what created those differences? Evolution created those differences. And so what I do is I try to unravel what created the differences. And so going forward, what do we need so that these differences are maintained in the population going forward? So, in other words, to prevent any more genetic diversity from going extinct than we already have. And so how do we maintain as much diversity? Because why is diversity important? It's important because if we don't have it, then we're all going to be the same, right? So if we're all the same, it's not good, because it means that if one disease comes through the population, everybody's going to get wiped out because they're all the same, or they're all genetically very similar. Which is why it's so important to have diversity in the population. Even though, you know, as far as we're concerned, one rhino looks the same as the next, but I can tell you one thing, some populations have a lot of diversity among their rhinos, and some, like here in South Africa, have very little diversity. And that's telling us that the populations went through various population changes back in time that now leads to the diversity that they have today. So the other way of figuring out how biological information is structured across the landscape is through genetics. And you can use two kinds, and I'm not going to get into too much detail with genetic jargon. And there's several ways of doing that. Let's just suffice to say that. If we look at the rhino, the black rhino is the most widespread of all the 5 rhino species. You guys know what the 5 rhino species are, right? There's the white and the black. I'll give you that clue. What else? Indian, Sumatran, and the Javan. So 3 from Asia, 2 from Africa. Okay? The 2 from Africa are both 2-horned. The Sumatran in Asia is also 2-horned, but the other 2, the Javan and the Indian, are 1-horned. Okay, so which is why our animal is called Diceros bicornis. Okay, 2 horns. So this is the range of the black rhino, and the first black rhinos appears in the fossil record in Kubifora near Lake Turkana in Kenya about 2.5 million years ago. So the modern form of the black rhino has been around for about 2.5 million years. The modern form of us, humans, does anybody know? How long has the modern form of human been around? No, 10 million, we still had it was still the ancestor with the chimp. About 300 to 600,000 years ago. So modern humans emerged out of the fossil record at around that time. So rhinos, black rhino, has been around for a whole lot longer than humans. Okay, when the black rhino came out, there was still Homo erectus running around Africa. There were still several species of upright walking ape man, Australopithecus and so on. Homo had just emerged on the fossil record, Homo habilis, and Homo erectus had only just evolved 2.5 million years ago. That was our nearest direct ancestor. Okay, so 500,000 years later, they appear in Langebandweg, and they appear again in Ethiopia 1.8 million years ago. So the species has been around for a long time, and what we see is that from East Africa, we generally have high genetic diversity. No matter where we sample black rhino in East Africa, we got high genetic diversity, and the genetic diversity tails off as you move away from East Africa. It's lower and lower and lower towards the extremes of the range, and that's not necessary. That's kind of normal for most species, and what we can say from that is because the place where we have the highest diversity is generally where the species originated. Okay, because those animals in that area have had long enough to evolve this genetic diversity compared to animals that are in the extreme ends of the range, which are more derived. So for humans, for example, where is our highest diversity? It's also in Africa, okay, which is why we also know that all of us originally come from an African stock, okay, because put it like this, the genetic diversity between a human living in Japan compared to a human living in Europe is less than between two random people sampled from a village somewhere in Nigeria. Okay? So those two randomly sampled Nigerians have more genetic diversity between them than a Chinese has, or a Japanese has, a human Chinese has to a human European. Okay? So the taxonomy of the black rhino has been in contention for decades and decades and decades. And these are the two schemes that we use. This is Colin Groves. He died now, unfortunately, a very famous Australian taxonomist. And he came up with these 1, 2, 3, 4, 5, 6, 7/8 subspecies of black rhino based on skulls. How the skull, he measured skulls and so on. And based on the differences in skulls, he came up with this scheme. Raoul Dutoit and his colleagues at the African Rhino Specialist Groups realized that this scheme was a little bit too complicated because by the time, so this is the scientists and here are the managers essentially, okay? And the managers decided, no, that's too complicated because the rhinos are not respecting political boundaries here. So let us go with this scheme where we say, okay, everything in Kenya is managed in a certain way. That's one subspecies. Everything in South Africa and Zimbabwe is managed a certain way. And everything in Namibia is managed a certain way. And so that divides the management into countries, which is, they call it pragmatic. But I have another word for it. The problem with, so this is how black rhino have been managed for the last 30 years. This is what we knew about the structure of the biodiversity. Okay, and so you can see already they're at odds with each other. As a result, we've had animals from Namibia. I don't know if you've ever been to the Cape. You've been to Addo. You'll only see Namibian animals, korkol and animals, black rhino because we all thought that the Namibian rhino extended into the Cape of Good Hope, which so if you managed based on this scheme, you would never have done that, right? Because you can see very clearly that Namibia is more closely related to this, so you'd never move animals down into the Cape. So the Cape rhino is extinct, right? But based on this scheme, it was quite fine to move rhinos down into the Cape of Good Hope from Namibia. So we got Namibian rhinos in the Cape. We've got our Zululand rhinos in the Luangwa Valley because the Luangwa rhinos were poached out in the 1980s. We've got Zululand animals in Malawi, in the Shiri Valley. I don't know if you've been to Liwonde or Majete. They've introduced Zululand rhinos to there. We've even got Zululand rhinos. When I say Zululand, I mean Umfolozi black rhinos. Okay, so all South African white rhino come from Umfolozi, and almost all our black rhino also come from Umfolozi. So Kruger Park, there was no rhino. They went extinct. Okay, they were shot out. So all the rhino we have in Kruger Park and everywhere else in South Africa are from KZN. Okay, which is why we call our animals the Zululand stock, but even though they're not necessarily from Zululand. They could be from Pilanesberg, but they're still Umfolozi ex-Umfolozi animals. So, and we've moved Zululand animals even into the Ngorongoro crater. And inside East Africa, in Kenya, so what happened in Kenya was also quite, it was not ideal. So little do they know they had two different kinds of black rhino in Kenya. Now, in retrospect, we know this after we've done all the genetics and so on. But actually, when they were trying to conserve black rhino in Kenya in the early 1980s, there were black rhino literally in the rural areas in dribs and drabs. You know, one here, two here, one here. And so the Kenyans realized that the game was up for black rhino in that country. If they just allowed the situation to continue, most of those animals would have been wiped out. So what the Kenyans did is that they gathered animals wherever they could find them, and they put them into about 3 or 4 what they call IPZs, intensive protection zones. And so unwittingly, what they did was actually they mixed 2 different kinds of black rhino in Kenya. Okay, so the 2 very distinct genetics are now mixed in East Africa. So we'll get to that. So yeah, the reason I have Mike Bruford's picture up here. Mike Bruford is a former mentor of mine. He died a couple of years ago, well before his time. But this is a project that I started when I was working with him in Cardiff University. And what we did was, because it's such a widespread species, we thought to do a definitive study, we have to go to pretty much everywhere where there were black rhino historically and get samples. And the way to do this is because, I mean, let's face it, there's not many places with this black rhino left. What are the countries still with black rhino? I mean, where they haven't been wiped out. There's South Africa. Where else? Kenya. Namibia. And there's only 2 others. Zimbabwe. No. Tanzania. Okay? Those are the only 5 countries in the world where black rhino have not gone extinct. They've gone extinct everywhere else. And everywhere else you see them, they've been brought in from one of these 5 places. Okay, 10 years ago, or 15 years ago, we had a 6th one in Cameroon, but that was declared extinct in 2011. So, all these black dots is where I obtained a black rhino sample. And you will be the first to say, well, there's none left in Angola. How did you go there and get black? Well, there's none left in Zambia. Well, there's none left in South DRC. How did you? No, these are all from museums. So luckily, you know, lots of rich European people toured Africa in their hundreds and shot everything that moved. So they shot everything that moved, and a small fraction of the things that they shot wound up in museums in Europe and in the United States. And this is actually where my field is. So inside a museum where I'm taking a sample from a rhino or whatever, and then with this museum sample that's been dead for 150 years, we extract the DNA, and then we can figure out what were the genetics of the rhinos that were there, even though they're not there anymore, they're extinct. Okay, so this is our sampling, and we came up with 1, 2, 3, 4, 9 different groups of black rhino. This was in 2017. And what we figured out was that the differences between these groups, there was always some major river or rift valley or something that separated these boxes, right? Like the Shari Lagoni or the Nile or the Zambezi. And so that was telling us that rhino genetics on either side of major rivers is different, or black rhino genetics. And that's telling us that they probably don't swim very well, right? You'll never see this kind of structure with elephants or buffalo. They are all similar to each other. There's no boxes with, Lake Victoria, Namibia, South Africa, Kenya, Tanzania. There are no boxes like that, like there are with black rhino. With elephants, it's pretty much all one thing. With buffalo, it's pretty much all one thing. And so that's because these animals swim. These animals migrate over rough terrain. What we're finding with rhinos is that they don't like swimming. They don't like crossing rift valleys. They're very similar to giraffe, believe it or not. Giraffe also don't like rough terrain. Giraffe also don't like crossing escarpments. There's no giraffe in the Zambezi Valley. Have you ever been to Mana Pools? There's no giraffe there. Why? There's no wildebeest there. So there's some animals that just have a rough time crossing either rough terrain, rift valleys, or river valleys, or deep rivers. Rivers that have been flowing for hundreds of thousands of years. And that's telling us also that these rivers tend to separate populations. It's also telling us that the river has probably been flowing now for a long time, because the river has to prevent animals crossing for the animal populations on either side of the river to evolve differently, right? Whereas if the river flowed and in 100 years it stops flowing, and then in 100 years again it starts flowing, you wouldn't see these genetic differences on either side of the river. If you see genetic differences, unless of course it's a bird which can fly across, or an animal that can easily swim across, but for something like a rhino or a giraffe or a wildebeest, it's actually, well, wildebeest swim quite well. But let's just focus on giraffe and rhino. They really are big differences between either side of the Zambezi, either side of the Tana River, either side of the Shari River. And so it's very clear that for some species that rivers, just a river that's been flowing for 100 or 200,000 years, can create a barrier that can't really be crossed, and you have the evolution of different subspecies on either side of that barrier. So this is what we found out with black rhino. So the results that I just showed you up here are from what we call mitochondrial DNA. It's passed on only through the females in the population, and so because it's only passed on by the females, it's the gene pool is much smaller. There's only one female is passing mitochondrial DNA to another female, so that's a 1-to-1 inheritance. Whereas if you think about the DNA you have in your nucleus, so the DNA you got from mom and dad, mom has 2 chromosomes, segregation occurred and you get one of them. Or they mix and then you get one of them. And your dad had 2, and the very same thing happened in him, and then you get one of them, and so you've got 2, you don't have 4. Right? So every 1 genome comes from a potential 4 genomes. So with mitochondrial DNA, the gene pool is 4 times smaller, literally, than with nuclear DNA, which is why we see these beautiful patterns with mitochondrial DNA, which we may not see with the nuclear genome. So what we did was, this is Fatima Sanchez-Birrero. She was a PhD student of mine who did this study. And she sampled, well, the very same places that I sampled with the mitochondrial DNA, except she obtained whole genomes from these animals. So a whole genome is basically all the DNA in that animal gets sequenced. And it's if you assemble an entire genome of a mammal, like you or me, it's around 3 billion base pairs from one end to the other. With birds, it's around 1 billion base pairs, so about a third the size of a mammal genome. But I mean, just think about it, 3 billion base pairs of DNA. So now there's nowhere for the rhino to hide. We know everything about these rhinos. And what I love the most about it is that they're almost all of them from museums. And when they were shot, nobody ever thought, oh, somebody's going to come along in 100 years or 200 years and sample them and get DNA out of them and do cool studies that could help their conservation. Nobody ever thought of that, right? So the fact that we can obtain this information from museum collections across the world it's sort of like the animal didn't have to die in vain just because some person wanted to shoot it 100 years ago. There's actually something of that animal is still there and can still help in the conservation of other members of its own species. So I think that's really cool. So Fatima managed to obtain 64 genomes from museum specimens, and what she found was that there are these 6 major populations across Africa. And I'm not going to bore us too much by just saying that, well, we'll see it on the map soon. We'll see it on the map soon. There are these 6 major populations, and here they are in different colors. So you have south of the Chobe Zambezi, you've got the southern African black rhino. North of the Zambezi, all the way to the Tana, you've got the East Africa, or the Central African black rhino. And then north of the Tana, you've got the East African black rhino. And what's interesting is between the Shiri, so between Lake Malawi and the Rufiji, in this southeastern corner of Tanzania, or northeastern corner of Mozambique, we have our own different kind of rhino, which we never knew existed before we did the genetics. And of course, we've got a distinct kind of rhino in the Horn of Africa, and another distinct kind of rhino in West Africa. So if we dig a little deeper into the structure that Fatima found, we found actually that if we dig a little deeper from the 6 population, so guys, I don't want to bore you with this, but this is what we call a structure plot. And I should have explained that to you beforehand, because otherwise it just looks like sort of Tetris, right? So each line here, so you can see that there are different lines, right? And the lines have different colors, right? So each line, so this one is one line, here's another. So there are 4 lines here which are brown, right? You see that, everybody sees that. So each line is a different black rhino. Okay, each line is a different black rhino. And the first 4 are from Chad. Okay, so the first 4 are from Chad, and that's a different color. So it's saying that genetically there are different populations. So the different colors means that there are different populations genetically. Okay, and you see some of these lines, for example, look at this one here. This is one rhino, but this line, the genetics is telling us that this rhino is a mixture of 3 different populations. Okay, this rhino is a mixture of this East African population, this red Northern Rift population, and this Maasai land, this Eastern Rift population here. So at some stage, the ancestors of this rhino were from different populations. Whereas at some stage, at no stage were the ancestors of these rhinos from a different population. So these rhinos are all pure individuals of that population, call it beige. Okay? And so this is how we read this plot. And so so you can see very clearly that there are animals with this solid colors, but there are many that have more than one color, right? So their genomes are made of ancestry from different populations. That's telling us that not only do these different populations evolve, maybe on either side of a river, but at some stage the river stopped flowing, allowing gene flow to occur, and more mixing to occur, which is why you get a rhino. These are all museum samples. So they're not taken from a zoo where you can say, oh, they brought an animal from Kenya, they brought one from South Africa, that's why this animal is mixed. No, these are all from museums, which means that they're naturally from the place they were shot. And so there's a lot of natural mixing in black rhino. So there's not only evolution because they become separated and they evolve differences, but then whatever was separating them is every now and then stops separating them, and they're able to breed again, creating animals with typically these kinds of profiles, okay? So if we dig a little deeper into the data, we see that there are actually 10 different populations. If you look at all these colors, in this last plot at the bottom here, there are 10 different colors. So we figured there are probably somewhere between 6 and 10 distinct populations of black rhino, historically. And so now that we know that we have these 10 different populations, what does that mean in terms of subspecies? So now subspecies, I have to tell you already that it's a word that I really hate. When I was a student, it was a big debate whether this thing even existed, right? And you can't get a politician to put money into saving anything if it doesn't have its own subspecies or its own species, right? I mean, you try getting a politician to give money to this really distinct population, or, as we call it, or or evolutionary significant unit, as another way of calling them. That doesn't work. But if you say subspecies, immediately people are like, oh, we have our own subspecies in South Africa. Oh, let's conserve it. Whereas if I said we have our own population in South Africa, people couldn't care less, even though it's the very same thing. So a subspecies, unfortunately, implies that there are two things, so two different subspecies, and one ends at some point, and the other begins at another point on the map. And a lot of taxonomists draw these things like that map I showed you earlier, but we don't really know what was the barrier between these species. What was it, a river? Was it a mountain range? What was separating these subspecies? Because it's within one species, what's not stopping them from just mating with each other at that place where they supposedly meet? Okay, and that's the problem with the subspecies. They're not a distinct entity. Because a species is a distinct entity. It can't breed with another species. Whereas two subspecies can happily breed with each other if they meet. Okay, so, which is why I prefer the word population, but nevertheless, we use this really cool traffic light approach to determine the number of subspecies that are in for black rhino. And this is the table we came up with. We came up with 9 different subspecies. You don't have to worry about it. I'll put it up on the map here. And that is the Cape rhino. So the animals from Cape Town are no different from the animals in Zululand or the Zambezi. Okay, so all of the animals along the east coast, southern South Africa, all the way to the Zambezi along the east coast are one entity. Okay, so and then we have got the Namibian entity, which is completely separate, genetically different to the South African and Zimbabwean black rhino. So again, now for conservation management, one can move Zimbabwean rhino into South Africa and South African rhino into Zimbabwe without any issues. If we're in South Africa, and especially in South Africa, we've got these small populations of rhino, and we want, oh, where can we find a male that's going to bring good genes into our population? Well, this is going to help you to figure out where to get that male from. Anywhere inside this blue here, if you can source a rhino, then that's a good idea. Okay, so just imagine now, moving these Southwest African animals into the Cape was actually not such a wise idea, right? Having Namibian rhino in Addo Elephant National Park is not a good idea, because that's not the natural distribution of things. Yep, so we've got our Namibian black rhino, we've got our Ruvuma black rhino in southeastern Tanzania, we've got our Central African black rhino, the Maasai Land black rhino, the East African black rhino, the Northern Rift black rhino, the Horn of Africa black rhino, and lastly the Western black rhino. So this is what we had with black rhino. What's gone? What do we still have? We still have Namibia, we still have South Africa, we still have Zimbabwe. So we've got 2 subspecies. We've still got Central Africa in low numbers, mainly in zoos. And we've got the East African kind in Kenya, mainly in Kenya. And we've luckily got the animals of the Masai Mara. That's a completely different subspecies again. So we've got these 5 groups of rhino that are still around compared to the 9 that we used to have. And given that, there were 100,000 black rhino in 1970, by 1992 there were only 2,500 left. Okay? So that's like, 90 or 98% reduction in numbers, and yet, in terms of the biodiversity, we had 9, and now we've got 5. Okay, so we may have lost 98% in terms of numbers, but we've lost just under half of the total biodiversity that we have for black rhino. And so now that we know what we had, what we lost, we need to figure out how best to conserve the few that are still remaining to us. Because these are gone, these will be gone, and unfortunately, I don't know who's been to Selous Game Reserve anytime recently, but there's no more black rhino in Selous Game Reserve. The Tanzanians will tell you there is, but they're not there. Like the Cameroonians told us they were rhino for 15 years before we realized they were actually not there. So, well, what does that mean in terms of moving animals, what we've done in the past? We've moved animals way outside the areas where nature would have had them in an ideal world. Okay, we've taken all these Zululand animals up here into East Africa, and in some cases have even mixed with the local rhinos. And so we've got this problem where you've got lots of different genetics mixing which would not have happened in nature. So one might say, well, who cares about that? If they're healthy, who cares, right? Let them mix. Yes, that's great. But remember, our goal is to conserve as much of the diversity that we have. So if, say, you've mixed two different populations or subspecies of rhino, and you say, well, they're already mixed, like in Kenya, for example. What can we do? Let's just let it be and keep them like that. Well, eventually what will happen is that there's something called genetic drift. And I don't want to talk too technically about this, but genetic drift is what we call a population effect. So if a population is large, the effect of genetic drift, a random event, is not very significant. So for example, let's put it like this. If you have a population of 100 and one individual dies in that population because they got struck by lightning, well, you still have 99 more. And the 99 probably should have all the genes that one who died had. So it's not a big deal. Whereas if you have a population of 10 and one gets struck by lightning, then maybe the 9 don't have the genes that one had. The risk is higher that you're going to actually lose unique genes when the population is small, as opposed to when the population is high. That's all genetic drift actually is. And so when you mix and generations go by and you don't monitor the population, you might have mixed two different subspecies, but 5 generations down the line, you do your genetic test and you find actually we've only got one, or one's genes are sitting at 95%, the other's at 5%. It's because the population has simply drifted towards one population and away from another. And now we've all but lost that population. And that's because we mixed them and didn't monitor what we were doing. So mixing can be a good thing, but mixing can also be a bad thing, especially if you're mixing two very different biological kinds of animal and you're not managing appropriately. So you don't know what you've got. You don't know who's breeding at each generation. There's no stud book. And in that case, then you really do run the risk of actually losing one of the two things you originally mixed. Okay, so basically what the study has entailed is a complete overhaul of black rhino conservation management. And so based on these studies, I get invited to talks all over the place to advise conservationists on the way forward for black rhino conservation management. So now, for example, in Kenya, where we've got both this Mikeni type and the Ladoensis type in mixed populations, the Kenyans, I mean, they see the work, they want to manage, but they don't actually know how they should go about it. And so, so doing the work, telling the conservationists, this is what evolution created, okay? So it's up to us, as this is what we have of what evolution created. Now it's up to us to see how much of this is still in our power to save while we can, because maybe in the 1970s or the 1980s, there was all this other diversity around, but we didn't have the power to save it. It's all gone now. And we can show them, these are the ones that are gone. And so, do we really want to continue in the same kind of way of managing that we've always done for the last 2 or 3 decades and continue to lose out the diversity that we still have? Just to give you an example, South Africa has got, they're now about 5,000 black rhino. Maybe 6,000 even. 2,500 of those live in Namibia. 2,500 of those live in South Africa. So it's 5,000 out of 6,000, right? So that's 2 subspecies with 5,000 animals. Compared to the other 3 subspecies that are still out in East Africa, one with 500, one with 200, and one with 50. Okay, so this is the idea about genetics. It's not a numbers game. We've got 5,000 black rhino, but actually, the diversity, most of the diversity, 3/5 of the diversity, is sitting in animals which are not even 1,000 animals. So the time has come to stop multiplying more Namibian and more Zululand rhinos and actually focus our attention where the numbers are actually lower and the diversity is highest. So that would be my recommendation, is to focus on East Africa. We're fine in Southern Africa for the moment. East Africa is where the cuck is hitting the fan, and where they've got so much diversity that they still can lose if they don't manage properly. Okay, I think I'll leave it there because otherwise we're going to get into too much genetics. So thanks very much.
Right, sorry, I don't have a microphone, but I have a very loud voice. So firstly, massive thank you. That was an incredible presentation. So round of applause again. Thank you. All right, I think we're going to have a short round of questions. If I can just ask if you could please repeat the question into the microphone, just so that there are any viewers watching the recording can hear that. So if there are any hands up with any questions off to start with, right there at the back, we got our first one.
Thanks very much. Great talk. Very insightful. How is this information being received by specialists from the management perspective going forward? And secondly, have you done a status quo? So what's the current population, current population?
So you mean the current population?
Current population, how much of that?
Yeah. Okay, so the question was, what was the reaction of all of this by the specialist group that manages black rhino? And the second question was, you presented the historical stuff, the museum stuff. What does it look like currently, this current status quo? Okay, so let me answer the first one first, and that was, well, how people react to evidence, how conservationists react to evidence is entirely dependent on what you tell them. So in South Africa, where we said, oh, what we've been doing up to now is just brilliant because we've been managing Namibia separately from South Africa. We've actually also been managing Zimbabwe separately from South Africa. Now what we've told them is, well, you don't have to do that anymore. You can manage Zimbabwe together with South Africa, which is going to be great for South Africa because South Africa has got very low genetic diversity, and we could really use an influx of some of those Zambezi genes. But what was great is that we're not managing together. So Namibia is kept separately. The Namibian black rhino is kept separately from a South African one, and that's what we've been doing, and everybody was quite happy with that. Then in East Africa, we highlight all the biodiversity and say, well, actually in Kenya, you've mixed these two. So in Kenya, whatever the current management practice is not actually what you ought to be doing. So now, as soon as you say that, the conservationists don't like it. Because now they have to actually manage. I do understand. I mean, just knocking down one rhino and moving them from reserve A to reserve B is an enormous task, let alone for 500 or more. So, and the last thing they want to is for some ivory person from an ivory tower like me coming there and saying, oh, look, this is what you guys need to do. Little do we realize it's going to cost, $3 million US just to do that intervention. And so I do appreciate all of there's a lot of pushback. So to answer your question, it depends what you're saying. In situations where the management that's happening is what you would recommend, more or less, we're fine. The problems come when the management that's happening is not what you'd recommend. So for example, in Kenya, they would have to, they've already caught every animal. They've already got DNA samples from every animal. They just haven't looked at them. So what they need to do is look at each animal, look at its DNA profile, and figure out which of those two populations it belongs to, and start managing them appropriately. Because if they manage them all as one big population, one of those populations unfortunately going to go extinct. So, right, your second part of your question was, what is the status quo now? So the status quo now is that we've got these 5. Okay? So we also, what I didn't present is we also have data from animals that are alive today, not just the museum animals. And they also show this patterning, this structure, as we call it. And so there are 5 left. There's Namibia, South Africa Zim. South Africa Zim is 1. The Masai Mara is 1. And then Kenya north of the Tana River and Kenya south of the Tana River. So southern Kenya and Tanzania is 1, and then northern Kenya, southern Sudan, Uganda is another. So yeah, there are these 5. But the Kenyans, they really don't want to hear it. They just don't want to hear it. And so these are real life issues to conserving animals. In fact, the major problem with conservation comes when you now have to deal with the human who's actually going to have to do the conservation, as opposed to doing the science. I mean, it's taken a long time to get this. I mean, to get that many samples from that many museums, man, I really had to travel around the world to pick up this sample set. And nobody appreciates that kind of field work. Really, nobody appreciated it. And then they say, well, your sample size is this is your sample size. You know, it's not, it's actually data. It's telling us something. And genetics, you know, unlike, this is why I really love history. I almost never read fiction. I'm sounding really terrible. I'm sorry. You're going to call me a geek after this. I almost never read fiction. I love reading history. But if you think about it, history is written by somebody. A human wrote that book, right? And somebody probably paid that human to write that book in that way. Whereas the real history is what are the mutations that randomly occur in the genome. And these are the things that we unravel to come up with pictures like the one I showed you about how the rhinos evolved and so on. That's because their history has been ticking away with mutation after mutation after mutation with time, and there was no agenda, right? Those mutations occurred at random. They got sorted randomly or through natural selection. And so that's, for me, is the real history. The history that is in the genome. The history that somebody didn't write. The history that nature wrote.
Sorry, I'm not going to say how I know, but the Rwandan rhinos.
Yes.
So they were supposed to be genetically Rwandan rhinos, even though they were kept in South Africa.
Yeah.
But there was a sneaky, apparently, I've heard, that the genetics weren't quite what they were supposed to be.
Oh, now you guys are talking about the real seedy world of black rhino conservation.
Because the Rwandan rhino...
I don't think many people know about this. Are you sure you want to air it out here? For the whole world to hear.
But so they were supposed to be purebred Rwandan East Africa.
Right, yes.
Which were worth double what?
Oh, more than double, mate.
Cash-wise, US dollar-wise, more than our rhinos were. However, what happened? Some sneaky South African rhino got in there. But they still took those rhinos there. But is there enough genetics left in that original Rwandan population to sort of bring those genetics back, or was it too tainted?
Right, so, wow. I was not expecting, that's a question from somebody who really is kind of deep into the black rhino, the murky world of black rhino politics. But yeah, okay, so thanks for bringing that up. That's the biggest scandal at the moment in black rhino conservation. And that is, just to give some background. So in the late 70s, when, as I was explaining to you, up in East Africa, there were black rhino in the landscape in the rural areas, but literally in dribs and drabs. 1 here, 2 there, 1 there. And so at that point, the Kenyans actually thought that they were going to go extinct. And so not only did they catch them wherever they could and brought them into these intensive protection zones and mixed them up unwittingly, like I explained to you, but a small proportion, like 6 of them, were caught in southern Kenya and moved, which is south of the Tana River. So it was only the one subspecies that they caught before the mixing happened, actually, funny enough. And so they brought them to, they put them in Addo Elephant National Park, where there were no black rhino at the time. And it was in the 70s and 80s, and they were supposed to be kept pure so that if anything happened in East Africa, we still have a reserve here, and we could breed them up and send them back to Kenya, and we'd still have East African black rhino. Unfortunately, the guys who were running the show in Sand Parks, while 2 bulls from Zululand were moved to Addo. So now you can just imagine now what a disaster, because the Zululand genetics is very different from the East African genetics. And so those Zululand animals, one of them didn't breed, but one of them was quite a successful breeder. And then when Sand Parks a few years later were told, hang on, that was a very, very bad thing. You've now brought South African rhino into this East African gene pool, and we are supposed to keep this East African gene pool in case things go badly up there, we can move them up to East Africa. So now Sand Parks realized their problem, and the people in charge thought, oh, we need to get rid of these bulls. So the one had already died, but the one that was successful, they got him out of there. And according to the people who worked in Addo, they had got every last offspring that rhino had sired. Okay, so Sand Parks had thought that they'd solved this problem. Okay, they'd put them in there, but now they got them out and it was fine. They're still pure East African animals. Until about, I don't know, about 6-7 years ago.
They sold them to private breeders in the Waterberg.
They sold them to various places. Yeah, they're actually in the water. They're based in the Waterberg. They're not based in Addo anymore. But the Akagera, they sold to about, I don't know, 20 or so animals to Akagera in Rwanda, where Africa Parks is just starting up, was just starting up that national park. And Rwandans, of course, had the East African type originally. So they wanted East African rhino. So they asked Kenya, and Kenya said, well, until we reach over 1,000 rhino in Kenya, we're not selling any. And so they were off the cards. Tanzania don't have more than 100 rhinos, so they were off the cards. And so it was just this one Addo population, which was now, I don't know, 70, 80 animals. Those 6 had bred up to 70, 80 animals. And so they thought, well, let's just get 20 from them. And so, but in the meantime, the scandal had broken. They'd already sold animals to various places. They sent one to the Ngorongoro crater. And now they suddenly realize, oh, about 1/3 of our entire herd of this so-called pure population is got South African admixture. So mixed with South African genes. And so now suddenly nobody wants these rhino because they tainted. And I mean, it's a bit of a disaster. So now the Akagera animals, if I'm not mistaken, it was before. So they were sent to Akagera after the scandal had broken. So by that stage, they now from this particular reserve, if they want to sell East African animals into East Africa, they have to have a genetic profile for each of the animals to show, okay, no, it is a pure Kenyan animal. Okay, we can send it. And they are so. With that, the problem is alleviated to some extent, but it doesn't explain what we do with the 1/3 of their herd. I mean, which is admixed between South Africa and East Africa, and a lot of people say, well, so what? They're rhinos. They're worth something, and I'm absolutely in agreement with that. If you consider where that mixture might have occurred in the wild. So the Zambezi River is the big barrier between the Southern African kind and the East African kind. So the Luangwa Valley in Zambia had the East African kind. But you can imagine that every now and then, because the Zambezi has been flowing for hundreds of thousands of years across that where it flows. But every now and then, I can't see a rhino not crossing over and not breeding successfully and creating what? Exactly that mix that we're seeing in about 1/3 of that admixed herd that nobody wants. So at the African Rhino Specialist Group, I also say, look, you guys, you can sell those rhino if somebody's starting a new reserve in Zambia, in the Kafue, or anywhere in Central Africa. It's fine to move those mixed animals into there. So don't just shoot them because you think that they don't have a conservation value. So yeah.
I won't even bring up chat, don't worry.
Yeah, we can talk about that. So yeah, it's clear that some people know a lot about what's going on in the black rhino world. Please, and there's no such thing as a stupid question, guys. I'm sorry about the genetics. I tried to make it as intuitive as possible, but if you have questions, there's no such thing as a silly question. Please just ask. Yes, yes. Yeah, so Solio has got both black and white rhino. In fact, I think it's the biggest population of white rhino in Kenya. So, but it's also got beautiful grazing, right? Which is not great for black rhino because they don't graze. Well, at least they certainly don't graze as a means of survival, whereas white rhino do. So in Solio Ranch, they've got like a mixture of the two black rhino subspecies. I'm not sure what the numbers of blacks are, but they've got at least 200 or close to 200 white rhino on that reserve. And so I think the way they can manage those carrying capacities is because there's just so much great grazing there. And a lot of people are going to say to me, well, what are white rhinos doing in Kenya, right? Because they're not naturally found in Kenya. They're found so where are white rhinos found? White rhinos are found in two places in Africa, or were until recently, right? And that is, so south of the Zambezi, Rhodesia, Namibia, Botswana land, South Africa. The only place you can still find them is Umfolozi. So in all of Southern Africa, they were wiped out, white rhino, and you could only find them in Umfolozi. Black rhino, similar, they were wiped out everywhere in South Africa except for Umfolozi and Mkuzi, okay? Whereas there's another kind of white rhino, right, that you know of. It's the northern white rhino. So our rhino here is the southern white rhino, and there's a northern white rhino that is on the, if you can take your mind back to the Great Lakes area of Central Africa, it's on the western side of the Nile. So along the Nile Valley and to the west, so into CAR and where's the other country there? Northern Congo, South Sudan, and so on. So unfortunately, the northern white rhino shares the same fate as many of these black rhino subspecies that have gone extinct. So there are only two animals left. There are two females that are post-reproductive, so they're too old to reproduce. And the last male died about 3 years ago in captivity. So that's it for the northern white. They're finished. There are two females left, but the subspecies, the population is extinct. And it's even too late to bring a southern white male to mate with those two females because they're post-reproductive. So unfortunately, they're gone. But white rhino, I mean, historically, when I say historically, I mean when people arrived during the colonial period, there were no white rhino in Kenya. There were no white rhino in Tanzania. But now if you go to these reserves, you find them. And when you do go on your East African safari and you see white rhino, and you feel, well, you know, how come an East African safari couldn't possibly be as good as a Southern African safari? Because it's our southern white rhinos that they have there. Okay, so they are from, all of them are from Umfolozi originally. So even though there are 200 running around Solio Ranch in Kenya, they're all Zululand white rhino.
Yoshan, what is the natural barrier between the northern rhino and the
I was hoping nobody would ask me that. But so there's lots of wildlife people here, right? So who's been to Central Botswana. Who's been to the Okavango? Right, loads of you, right? So who's been to Makgadikgadi or to Ngaipan? Okay, so on the way up to the Okavango, you got to cross this sort of area where there are a lot of salt pans. And the road from Nata to Maun crosses on the north is the Ngaipan and the south is the Makgadikgadi pans. And those of you who know that area can try to imagine that until about 10,000 years ago, that was an enormous inland sea. That was an enormous inland sea. Why do you think the Okavango has a delta into the Kalahari? Because originally it didn't have a delta into the Kalahari. Its delta was into this inland sea, okay, which was, we call it Paleo Lake Makgadikgadi. So that stretched from where the Okavango Delta is all the way south to even further south than Suapan. So basically all of central Botswana was inundated with this enormous paleo lake. And that no rhino could possibly cross that. And that paleo lake was from about 500,000 years ago till about 10,000 years ago. So you also, some of you may also be aware that, you know, if you've been up into, who's been to the Caprivi, so north of the Okavango, who's been to the Caprivi part of, okay, you've been to the Caprivi, and you'll notice that if you drive along the Caprivi, if you're driving from, it doesn't matter in which direction you're driving, east to west or west to east, you cross several major rivers. If you're driving from west, from Grootfontein, so from Rundu to Katiba Mulilo, so you're driving west, so you're driving east, you will first cross the Kavango, then you will cross the Kwando, and then you will cross the Zambezi at Kazangula. Okay? So there are these three enormous rivers, all of them coming from Angola, which is the north, coming south, and in the Caprivi, all of them do three weird things. The Kwando, well, firstly, the Kavango has a delta, like which river has a delta in the middle of the Kalahari. And then the next river down, the Kwando, has a dog leg, which is now called the Linyanti Swamps, and then it joins the Zambezi, which also has a dog leg. So it's coming down from Zambia, and then immediately turns and goes over the Victoria Falls. That's not the natural course for any of those three rivers. The natural course of those three rivers was straight through central Botswana, joining into what is the Limpopo drainage, and coming out of at Shai Shai, at the Indian Ocean. All of those rivers, that's why the Limpopo Valley is so enormous. You think, well, why do we have such a small river and such a huge valley for the Limpopo? From the Soutpansberg all the way across into Zimbabwe is that Limpopo Valley. It's an enormous 200 kilometers across, and yet the Limpopo is actually quite a small river. It's because those three major rivers, the Zambezi, the Kwando, and the Kavango, all of them drained their water. They were major rivers running straight through the middle of Botswana and draining out into the Limpopo Valley. And so about 500,000 years ago, there was a tectonic a shift, an earthquake. The plates only need to move a few centimeters. And what happens, these three rivers now were not flowing downhill anymore. So they couldn't get through central Botswana, and so they welled up in this enormous paleo lake. So these three rivers created this paleo lake, Makgadikgadi. And then eventually it overflowed to the north, and the Zambezi and the Kwando did what they're doing now. But the Okavango still had a delta into this major lake. So when the lake disappeared, that's why you still have the Okavango delta into the Kalahari. So it was this enormous lake there, and that's what was separating our Namibian population of black rhino from the South African stroke Zimbabwean population.
The Limpopo used to be the Zambezi.
Yes. Yeah.
All right, there's one last question. Hands. All right, fantastic. Can we get a last round of applause? Thank you so much.
Thank you.